My hope is that for most of you, this lecture is a bit of a review from things that you learned in your undergraduate and maybe even have gone over in some graduate courses. However, I've learned from experience that that's not always the case, that sometimes students miss this or have forgotten it since they've had it at an undergrad. So bear with me. If it's review for you, that's great. Remember it, reinforce it. If not, well, that's been my experience that some people this will be new for. So, population genetics, in broad terms, is the study of patterns and diversity at the population level. Most of what we've been talking about so far has been at the level of looking at species and relationships between species. So now we are going to instead focus on looking at patterns of diversity within a single breeding population, or you can, if you want to, for now we can replace that idea of a population with a species. And because of our ability to gather data, we have vast amounts of resources and data that we can use to analyze this, and new insights into patterns that we have suspected and have known about theoretically for a long time, but now we're able to investigate in great detail. So this is a diagram that you've seen before. It's this simple idea of a single population at the beginning with no diversity. So each dot represents a different allele, and the colors each dot represents an individual gene or allele in an organism, and then the colors represent when there's been a mutation, so we may define that as a different allele. And so we start with a completely homogeneous population for this gene, but then there's a new mutation. And that new mutation begins to spread throughout the population and eventually replaces the older variant. So this might be a sign of natural selection. Now, we'll talk as we get more complex about how we might assess whether or not there is natural selection going on here. And it's going to depend somewhat on how fast this new variant spreads, what the population size is, what are the chances that it might become more common just by chance, and so on. Okay? And so notice that we get a third variant that pops up before the blue one becomes totally fixed. So even once we lose this light blue variant, we still have some diversity in the population. And then we get speciation events occurring, right? And so on down the line. Now, the idea of being able to trace all of the modern genetic variation in a population back to a single point is called coalescence. Okay, so coalescence in some ways at the population level, you could think of it as speciation in reverse, right? Looking backwards to that point where we have speciation. But in a narrower sense, if we're talking about genetic diversity, coalescence is really looking at all of the alleles, or maybe more specifically polymorphisms. And if you don't know the difference for now, that's fine. We'll define it more succinctly here in a little bit. But looking at all of the genetic diversity for a gene in a population and tracing that back to a single genetic ancestor. And so even though historically there might have been other variants that arose and that have now gone extinct, we're really just looking at what occurs here and going back to a common ancestor. And so with the data we have in our modern populations, we can say a lot. We can't say everything because we're missing some of the genetic diversity. And unless we have historical samples of that population, we wouldn't know anything, for example, about this green allele or this blue allele because we'd only have the other ones represented in the population. Now notice, because of the patterns of new mutation and then coalescent, that all of the species in this diagram, no matter what allele we choose, share a common allelic ancestor. So there's the common allelic ancestor for all of the diversity present in population C, species C. There's the allelic ancestor for all of them in species B, and there it is in allelic A. So all of these have a common ancestor that is unique for that species and defines that species. And this is most often the case. This is why for for phylogenetics, we often don't need to worry much about the allelic diversity because coalescence happens quickly enough that we get unique and distinct signatures for each species. Now, let's go back just a little bit farther. Imagine that we sample the population here, where I've got my arrow. So there are 3 pink alleles in species A and 2 green alleles. Now, we still have the green allele in species B, which was inherited from this common genetic ancestor. And so some of that ancestral genetic diversity is represented in both species A and B. And then let's say that we sampled along that, let's just say that we sampled the blue one. Now notice that if we use that, we would trace a common allele ancestor for the green allele in species A. If we sample the green allele and one of the green alleles in species B, we would find the common ancestor of that allele right there. But if we sample the blue, or for that matter, any of the alleles in species C, we would trace their common ancestry, and eventually it would have a common ancestor with this green allele, but not until we get all the way back here. And so if that was our sample, that would be a misleading result. Even if we accurately reconstructed the gene history, we would say that species A and B are more closely related to each other than species C. And we talked about this. This is one of the confounding factors in phylogenetics, although it's not really common. And in fact, we see that if we sampled modern ones in this example, it wouldn't be a problem. But the ancient ones, if we sampled it a little earlier on, before coalescence had time to completely fix alleles in a population, it would be an issue. Now, we called that lineage sorting, and it's just one of three major confounding factors that can make gene histories different than species histories. All right, so lineage sorting is one of them. This is a review, hopefully you remember it. But what are the other two? Think about it. The other two factors that can confound our phylogenies, and even if we construct gene history correctly, we don't recover the species history. Okay, so the other two. Number one we talked about is lineage sorting. Number two is ideas of paralogy and orthology, right? If we accidentally sample paralogous genes, we might be tracing gene history to a deeper node that does not reflect the species history. And then the third one that can be a problem is xenology, right, or horizontal gene transfer. And typically those are rare enough in eukaryotes that we usually don't have to deal with them. They're exceptions to the rule. So if we have a large enough data set with multiple genes represented, they're not going to confuse our phylogenies. And in fact, we might be able to point those out because of the handful of genes that are disagreeing with the vast majority of genes. Now, what factors determine the average rate of coalescence? Critical for this is some basic assumptions, kind of a null hypothesis, if you will, to start with. The problem is that natural selection is going to play a very strong and a very complicated role in coalescence. So for instance, if this blue allele is really beneficial compared to the light blue allele, very soon it will be fixed because of natural selection. And if environment changes during that process to make another allele, maybe the blue allele becomes more, we might even get a reversal of a trend. So, although we can evaluate them with large data sets and fairly complex analyses, we can evaluate the impact of natural selection on population. It's difficult to draw general rules because it's really on a case-by-case basis. It depends on the alleles. It depends on the advantage conferred by those alleles. It depends on the environment and the stability of that environment. It depends on the other alleles and maybe genetics and genetic interaction of those alleles. So it gets very complicated very quickly. However, if we can disregard the forces of natural selection, then it becomes a little bit more simpler. There's still some stochasticity, or what we would call unpredictability in it, if we're looking at it on a case-by-case basis. But as a whole, if we look across many different genes, the genes that are not subject to natural selection are going to show general trends, and we are going to be able to make generalized statement about those genes. So that is basically a overview or a quick kind of a shorthand of neutrality, right? That genes that are evolving neutrally are not subject to the forces of natural selection. They are still subject to the forces of mutations, represented by the mutation rate, and they're still subject to the population size, although for some factors, population size cancels itself out. So, in reality, the average rate of coalescence depends primarily on the population size, but that can be offset by other factors. So mutation rates, which bring in diversity, are proportional to the population size. Which of those alleles become fixated in the population is inversely proportional to population size. So both mutation rate and population size play a role in the average rate of coalescence, right? Because if we have a gene that's coalescing and then we have new mutations popping up, that stops the coalescent process, right? If we have a very small population size and so we're losing a lot of allelic diversity very quickly, that is going to impact the coalescence. So in general, the higher the mutation rate, the slower the coalescence process. Or basically, you can think of it as those new mutations interrupting the coalescence process. The smaller the population, the faster the coalescence process occurs. Now, as we are studying these patterns, we have great data sets to look at. We're going to look at a couple specific examples, and then especially in our next unit, we're going to begin to draw more general conclusions. So let's just look at a special example quantitatively a little bit, but mainly qualitatively, kind of looking at it and explaining an example. ALDH2 is a protein that helps us to metabolize alcohol. There are a number of other proteins in the pathway, but this is one that's kind of central and very critical. And it turns out that in humans, there's a diagram of what that ALDH2 protein looks like. It turns out that in humans, there's some clear, distinct patterns that are based on your geographic history, where your ancestors came from. So let's look at it. And this is from large data sets that we're selecting. This is one that's been known for a long time. This is not any new recent news. But if we wanted to figure out any patterns, we might count the alleles. And so we might even subdivide the population geographically. So people of German origin, people of Finnish origin, so these are European, we have Papua New Guineas. Hungarians, Eastern European, right? Moving into Europe, into Asia, and then to farther Eastern Asian populations. And notice that the ALDH2 star 1 allele, right, which is one of the alleles, which is kind of what we might call the wild type allele. It works good. It's by far the most common allele in the human population. That it's at 100%. Basically, every person of German ancestry you can find is homozygous for this ALDH2-1 allele. But there's another allele, and this allele actually is much less efficient at metabolizing ethanol. And so people that have one or even two copies of this allele have trouble metabolizing ethanol. They do it a little bit more slowly. If they drink, right, you learn maybe even back in high school that you can metabolize, depending on your body weight, more or less, about 1 ounce of alcohol an hour. However, people with this ALDH2 star 2 allele, right, they have a much slower metabolic rate. If they are homozygous for that allele, then they have an even slower rate, and they might get, not only do they stay drunk longer, but they also show more of the bad side effects, the hangover, the headaches, those negative consequences of drinking alcohol. Now notice, we could say fairly clearly where this new mutation, where did this new allele arise? Well, it's in the Far East somewhere, maybe in China. Koreans and Chinese share a fairly recent common ancestor. There's a little bit of movement of this allele into other parts of Asia, but historically it didn't make it into Europe. Now realize this is historical data. Today this might change because of immigration, because of a lessening of migrations of barriers to immigration, right? It used to be that very few individuals from China made it to America. There was some immigration in the 19th century that increased the genetic diversity of the U.S. population, and individuals of Chinese ancestry began to migrate in, right? And so often when we do this, patterns of migration are important. And if we see clear geographic correlation between certain alleles and where those alleles are found, that's a really good sign that not only did that allele originate in that area, but migration back and forth between other areas is fairly limited. If historically, thousands of years ago, Koreans or Chinese individuals migrated into Europe and reproduced and left behind offspring, or vice versa, if Europeans migrated into China, then we would see maybe different patterns here. And so this is the study of demography, right? Demography in general is a study of populations, and it applies to population genetics, which is using a genetic side to study populations. Okay, and so we can say a lot about populations by adequately sampling modern diversity. Now, I want to give you a definition of this term we talked about earlier, polymorphism. And I'm going to use these very specifically and with intent, but realize that you might come across people who have slightly different versions or just don't understand the concept enough to make the distinction. So we already made the distinction early on between a gene and an allele, right? So we think of a gene as a region in the genome that has a specific protein product, although some of them are ribosome encoding or other things, but most of them are protein coding. That's the region, that's the gene. And then if we have different versions of the gene, we call them an allele, right? So you have a site on one of your chromosomes that helps determine eye color. And you might be homozygous or heterozygous for those alleles, and those get together depending on the genetics, and they determine your eye color. So hopefully the idea between a gene and an allele is very clear in your head. And I make that and I spend time reinforcing that point because I always see, sometimes even in publications that should know better, I see people using the term gene when what they really mean is allele. So if we look across the human population and say, oh, why are people different? And someone says, oh, because of different genes they have. That's not correct. And barring some really rare mutation, everyone has the same genes, right? It's just what alleles we have for that gene. So really it's more, we're different as a species because of allelic variation, and also there's environmental input, right? Nature versus nurture. But the genetic component that makes up the diversity of the human population is due to allelic diversity, right? Not gene diversity, just to be clear. Now, a polymorphism is a broader term. Polymorphism also includes alleles, but it can refer to genetic changes and mutations that we wouldn't count as an allele. So for instance, we have 3 major proteins on the surface of our red blood cells that make up our blood type system, right? The ABO blood typing system. And if we had a mutation, let's say in the O allele, but it didn't change it functionally, it was there, we could measure it, but there was no functional difference, we wouldn't call it a new allele. It's still O blood type. It would still react to the protein test that we do for blood type the exact same way. There wouldn't be any phenotypic or physiological difference, but there would be real genetic difference. So some polymorphisms, the ones that do not create a physical difference, right, that don't impact the phenotype, those are just polymorphisms. But some polymorphisms do impact the phenotype, and we call them alleles. So just to be clear, polymorphism is a more inclusive term that includes both alleles and genetic variants that do not impact the phenotype. And so we've used these for decades and decades, and fruit flies were one of the first groups that we did it. We looked for polymorphisms that made a physical difference because they were the easiest ones to measure, right? It's easy to look at a fruit fly and say, oh, look, this one's got a mutation that is making its eyes white, where the one down here looks more like what we would call the wild type. And so originally we tried to distinguish genes when we were doing this work that were not impacted a whole bunch by environment, because that just made it complicated, right? But you should know your genotype, what a genotype definition is, and that genotype contributes to the phenotype, but many phenotypes also have an environmental component. And so if there's no environmental component at all, then all we need to do is look at the physical features, and we know exactly kind of what the allelic levels of polymorphism are, right? So I could count how many white-eyed fruit flies, do a little bit of math, which we'll look at with the Hardy-Weinberg, and figure out how common this white allele is in the population. But have I captured all of the polymorphism? So what I want you to realize is that these early studies that were focusing just on the physical traits that we could see and measure, they were incredibly important and led to major breakthroughs in understanding, but they really are only looking at the tip of the iceberg. If we want to measure really levels of polymorphism, we need to do some DNA sequencing, because there's going to be even levels, even all of the red-eyed fruit flies, they may have different versions of the gene that are polymorphic, but do not impact the physical phenotype. So to really get a good handle on polymorphism, we need to sample, do genetic sequencing, and not only that, we need to do it with an adequate sampling of the population so that we are able to capture most, or at least a good statistical view of all of the polymorphisms in a population. Okay? So today, the method that we use is DNA sequencing in comparison of a large number of a large sample of the population. And it's getting faster and cheaper, so we often have very, very good data sets. Okay, so review, this is very fast, I'm going to do it quickly. Hopefully this is Bio 1 or maybe even high school biology for you, right? You now know what an allele is. We often represent them with these big A, little a letters, or you'll sometimes see A1, A2. I like the big A, little a system, big B, little b. We tend to choose letters that have a different uppercase and lowercase letter, just so they're easy to distinguish. Right? And so when we see this, this means that one allele has a physical impact that's different from the other allele. And although there are exceptions to the rule, most eukaryotes are diploid, and so we're only looking at two alleles in a single individual. If you have two copies of the same allele, of course, you're homozygous. If you have different copies, you're heterozygous. And we can then have these interesting genetics. Now, we're not going to get into all the details of genetics, but you do need to understand the basics. Dominance versus recessive, right? This example in the the geckos here, is an example of incomplete dominance. In this example, if you have two of one allele, you have this nice brown and yellow striping. If you're homozygous for the other allele, you have this white and brown. But if you're heterozygote, you have a completely a different phenotype. So these two get together and make something different when they are together. So make sure it's clear in your head. We have codominance, like the AB blood type, where both traits are expressed simultaneously. They have both antigens. And we have incomplete dominance, where there's some intermediate phenotype if you're a heterozygote. Okay, so just be aware of simple dominance, recessive, codominance, and incomplete dominance. Okay, and we're not going to get too much more complicated with that with genetics. We'll have a few other things that pop up here and there. Okay, so I'm going to add to this list. We have codominant, and then let's just add to it here quickly. I don't think I have the incomplete dominance on here. So make sure you're aware of that in your notes and make sure you know the difference between codominance and incomplete dominance. Okay? All right. So initially when people started to do this population studies quantitatively, they would look at one gene and they would count the alleles in the population. And again, early on, this was done with nice, easy traits that we could physically measure. And then as we advanced, we were able to do it genetically also. Okay? So homozygous for one gene, we'll call this the wild type, big A, big A. Let's say we start with a population and there is no genetic diversity at all for this gene. Everyone has big A, big A. We could figure out the frequency of the individuals that have big A, big A. Now, frequencies are expressed between 0 for 0% and 1 for 100%. Okay, so if you see 0.5, excuse me, if you see 0.5, that's a frequency of 50%. But the math works out better if we do numbers between 0 and 1 rather than percentages. Okay? We could count up all the heterozygotes if there was a mutation and a new allele was introduced into the population. And so time moves on, and soon we get another allele at some frequency in the population. So we could represent these with these little f and then subscript big A, the frequency of the heterozygote, and then the frequency of the homozygous recessive. Okay? Now, these are genotype frequencies, right? These are going to be important, and we'll come back to them. But we do not usually begin with the genotype frequencies unless we have a large data set. If we have a large data set, we can count the genotype frequencies, but the math itself begins once we take these genotype frequencies and turn them into allele frequencies. And so we're going to be reviewing Hardy-Weinberg, and you need to be familiar with it. Now, historically, traditionally, one of the alleles, usually the dominant one, if there is a dominant one, was designated by the coefficient lowercase p. And the other allele, the mutant version, if you will, was designated by q. But really, this is just convention. Okay? So the frequency of the big A is p. The frequency of the little a is q, although if you really wanted to, you could switch those. But one allele is just, and these are just letters to stand in our equations to represent the frequency of those. If we had no genetic diversity in the population at all, then the frequency of one of these alleles, p value, for instance, would be 1, and our q value would be 0. But if we're have genetic diversity for this gene, we're going to see numbers between 0 and 1, and if these are the only two alleles, then those two numbers together would sum to 1, representing 100% of all of the alleles. Okay? So, let's do a practice. Let's say you go into this gecko example, and you count 200 individuals, assuming that's an adequate size to represent the whole population. Maybe not, but if we did it randomly, it's probably a pretty good estimate. You count 50 individuals that have this genotype, 50 individuals that have that genotype, and in this case, remember, phenotype is indicative of the genotype. So we know immediately when we see this gecko that he's big A, big A. We know immediately this one is heterozygous and this one is homozygous for the other allele. And so we could do it just by looking at them. So these incomplete dominant examples are good ways to count alleles without doing any DNA sequencing. But we could do DNA sequencing and figure it out also. So we get these numbers. Now, I want to go through a little bit of an explanation of the derivation of where we get our allele frequencies. there are two ways to do this, and I usually teach a little bit of a less mathematical approach in my introductory biology, and this is the mathematical approach. If we had genotype counts like this, we could figure out the frequency of each of the alleles. And to do this, to figure out the frequency of the big A allele, we would start with the frequency of the homozygous dominant, right, which in this case is 50 out of 200, or 25.25, right? And then we would add in half the frequency of the heterozygotes. In this case, our heterozygotes are also 50 out of 200. So for this example, and we'll put it in here. For this example, and I'm not going to bother to do the math. We'll have to make this upper, do this. It's going to bother me if I keep it as subscript. Okay. We would say, oh, I'm going to add in 0.25. Got to make all of this. Okay. So I'm going to add in 0.25, representing the frequency of the big A allele, and then I'm going to to that add in half of the heterozygotes. So in total, we get 37.5, or 0.375 for that big A allele. And we know that just by looking at our genotype frequencies. Now, the way that I teach my students, which I think is a little bit more intuitive and a little mathematically easier, my undergrad students, if you want to do it this way, that works fine also. The other way you can do it is by counting up all the big A alleles and then just dividing by the total. So we have 50 individuals that carry 2 copies of the big A allele, right? So that would be 100 big A alleles. And we have 50 individuals that carry 3 copies of the big A allele, right? And so we would have 150 big A alleles out of a possible 400 alleles, right? And so if you do, what's 150 divided by 400? And if you've done it correctly, you should get 0.375. And so we're going to follow the same pattern here. Okay, so let's make sure we got this to superscript again. Sorry, out of subscript. Okay. So for the little a, we have 100 out of 200, or 50%. So 0.5. We're going to add in the same number. So for the heterozygotes, it's the same, right? 1/2 of 0.25, or 0.25. Let, I guess, be consistent whether I decide to do it with a 0 in front or not. And so we get a final of 0.625. Right? And again, if you've done this correctly, if we're only evaluating 2 alleles, these numbers have to sum to 1 because we're evaluating 100% of all of the alleles. Okay? Now, if you did the same thing, you could just add up all of the little a alleles. We have 100 individuals that carry 2 copies. That's 200 little a alleles, plus another 50 individuals that carry 1 copy, so 250 little a alleles out of 400 total, and you'd get the same number. So whether you choose to use the equation or whether you choose just to add up all the alleles and divide by the total, you'll get the same numbers and it doesn't matter. But you need to be able to take a genotype count and turn it into allele frequencies. Okay? So we've got our allele frequencies here. Now, that's the first step. But the next step is more of a theoretical jump, and we're going to explain it in a little bit of detail, but you may be already familiar with it. We'll talk about the why. 2 researchers, an English and a German fellow, realized early on that they could use these genotype counts to tell whether or not a population was evolving. And if it wasn't, they called it equilibrium. And so today we call it Hardy-Weinberg equilibrium after those 2 researchers. So we can take these numbers, which give us an allelic estimate of the population, but we can do an equation to determine whether or not they're in equilibrium. Are they in balance? And if they're out of balance, then we know that there's an evolutionary force that's acting on the population. Now, what Hardy and Weinberg realized that taken as a whole, alleles could be treated as independent events. So the chance of one allele getting next to another, getting paired with another allele is simply, unless I'll add it to this here. We've got it at the bottom. We'll talk about the y here. It's already done here, but the chance of getting it is the union of those probabilities. So we take the frequency of the big A, times it by its own frequency, and that's the chance of having a big A, big A. It's like if you had a bucket of red and white marbles, and you said half of the marbles are red and half of them are white. And you say, what are the chances of you picking 2 red marbles in a row? A red and then a red again. And you'd say, well, the chance of getting the first red is 0.5 or 50%. And then the chance of a second one is 0.5. So I do the union of those two. The probability of getting 2 in a row is 25% or 0.25. Okay? The probability of getting a white marble would be the same, right, since they're at equal frequencies. So we could do the same thing with that q squared. Now, the probability for the heterozygotes is a little different because you can get a heterozygote 2 ways. You can get a big A from mom and a little a from dad, or vice versa. So that's why we need to do the union of those 2 probabilities, and then we need to times it by 2. So it's the probability of P, the big A allele frequency, probability of Q, the little a allele frequency, and then we times it by 2 because it doesn't matter whether mom or dad donates either allele. All right? So that's the reason. It's simply a rather simple probability prediction. Now, what they realized, and this was the breakthrough, and I'm going to put the equation here, is that taken as a whole, we could predict what we should see from the population. Okay? Now, if you've calculated your P and Q correctly, then these numbers will always sum to 1. I'm just going to copy and paste. It'll be faster than doing the superscripts and subscript. So P squared plus 2PQ plus Q squared is going to equal 1. Oh man, they keep doing all the superscript, subscript stuff. I think with our modern computers they'd be smart enough, but oh well. Oops. Let me fix this or it's going to bother me. Okay. I think that plus is. Okay. So those numbers will always equal 1. And this is a very common mistake for undergraduates to make, is they'll plug the P and the Q value in, they'll see if it equals 1. If it does, they'll say, oh, everything is in equilibrium. But that's not the way to test it. So let's go back to our original numbers here that we did, right? So we figured out a P value and a Q value for this gecko population. And let's plug these into our equation, okay? So we would have, oops, got to go here, 0.375, right, squared. And that becomes a prediction for what our homozygous dominant genotype should be. If everything is in balance, then this should be 0.375 squared should equal it. So let's just do that one. I'll let you guys do the other one, but you can really test it with any of them. If one of our predictions doesn't match the original observation, then that means it's not in Hardy-Weinberg equilibrium. So 0.375 squared. Let's just do that quickly. I'm going to use Excel. You could use calculator, however you want to do it. Oh, in fact, I've already got it calculated there. Oops. So I could simply do that one times itself. And so we are predicting 0.1406, or about 14% of the population should be homozygous dominant. That's what the Hardy-Weinberg equation predicts. And when we come back to it, we looked at the original population, it's 50 out of 200. It's actually 25%. So Hardy-Weinberg predicted only 14%, but we see 25%. So what this means is this population is not in Hardy-Weinberg equilibrium. There's something off with it. Now, if you didn't follow all of that, hopefully, again, it's review 4. If you didn't follow it, the next lecture is going to be practice and then looking at specific patterns that we can see in the Hardy-Weinberg equilibrium. So don't freak out if you're still not okay with it. If you're, wait, how did Dr. Terry get that? And you could check the 2pq value and the q-squared value. Just hang on, look at the next unit. We'll get some practice. I'll give you some examples to work out on your own and pause the video and then come back, okay? But again, if you are given a genotype frequency or genotype counts, you should be able to take those and using either the equation or counting all of the alleles and dividing by the total, you should be able to give me allele frequencies. You should then be able to take those allele frequencies and put them into the equation and determine whether or not that population is in Hardy-Weinberg equilibrium. And if it is, all of the numbers, the p-squared will match your original observation. 2pq will match your original observation. Q-squared will match the homozygous recessive in your original observation. Okay? If they don't match, then we're not in equilibrium. So what does it mean if we're not in equilibrium? Well, it means there's some evolutionary force acting on that population. And so here are possibilities. I'm going to go through these quickly. Now, we're making the basic assumption that this is a diploid sexually reproducing population, okay? It only works. Otherwise, you can modify the equation if you're looking at triploid or if you're looking at more than 2 alleles, but we're only going to do a 2 allele, so it's always p-squared and q-squared, okay? And then the remaining here, 2 through 6, show the things that need to be in place for it to balance out. There needs to be completely random mating, right? Because those association of those alleles needs to be randomized. If there's not, it'll be out of balance. There needs to be no new mutations. If there are new mutations, it will be out of balance. We need to make an infinite population size. Now, in reality, no biological population is infinite. Some are very, very large. But we get to a point where even finite population sizes, if they're big enough, approximate Hardy-Weinberg equilibrium. So if you want to put in your notes or put in parentheses large population size, that works fine. There needs to be no natural selection. We'll look at some forms of natural selection, the sign that they leave in Hardy-Weinberg, and then there needs to be no migration. So if all of these things are met, then your p-squared value that you calculate from your allele frequency will also match your original observations. Okay, and I'm going to do examples on the next unit, so if you're still a little lost, that's okay. Okay, now, also I will derive an equation for 3 alleles, and then if you want to, you could do more, but the probabilities are rather simple once you figure out. So I will do an example with 3 alleles at the beginning of the next lecture.